If you please come back and have a seat, everyone, please. I'm gonna put my phone. There are lots of empty seats at the front. And if you don't have a program, there's some at the back. You can just pick one up. It'd be nice if we filled all the seats in sequence. Okay. There are some people who will be joining very soon because they probably don't want to be in the posters, something like that. But thank you everyone for coming. And I like to make a little bit of a noise. So just to start the event, we will give a very big clap. Okay, round of applause at the count of 3, just so everyone knows we're here and they can leave what they're doing and come. Okay, 1, 2, 3. Okay. So for those of you who weren't here, I'm Linda Oyama, and I'm very excited to be putting together this event with all of my colleagues as part of the Queen's AMR Hub, so Antimicrobial Resistance. So all of the researchers, some people who I've never met before, so it's very exciting. We're going to kick off the event with our first talk from Dr. Simon Doherty, who is a staff with us here, a staff member here, a senior lecturer, and Simon is a vet and was a former president of the British Veterinary Association. I'm not going to say too much. He's going to tell us about One Health and AMR. Thank you, Simon.
Thanks so much, Linda, and thanks so much for organising this event today. I think it's it's absolutely fantastic to be able to kind of showcase what we're about here in relation to our work in and around AMR. You'll probably gather as I go through my presentation. First of all, disclaimer at the start: I am not a traditional academic. Okay, I'm a vet by training. I'm very much a clinician, and my work here at Queen's is very much about feeding into other people's work with hopefully some practical aspects of veterinary medicine and how it applies in relation to the field, and that obviously includes antimicrobial resistance. So the title for my talk is just One Health: Opportunity through Collaboration in Tackling AMR. Some of this is going to be, for this audience, is going to be teaching Granny to suck eggs. It's back to basics. What is One Health and how that relates to AMR? But maybe just a couple of personal reflections then, sort of coming through from my perspective as a practicing veterinary surgeon as well. So I'm delighted to be now officially part of Linda's lab as well. So we've got the microbiome, antimicrobial resistance and One Health lab here at Queen's, and I'm delighted to be able to play a part in that as well. So a few years ago, I suppose a lot of people would have thought about One Health as being this kind of slightly ethereal, slightly fluffy, up in the clouds, kind of very strategic, people, animals, and the environment. This interface between people, animals, and the environment. But it is an awful lot more than that. It's not a new thing. We go back to Hippocrates. He said, if you want to learn about the health of a population, look at the air they breathe, the water they drink, and the places they live. So 2,500 years ago, there were links between the health of people, animals, and the environment. Pasteur, every time I throw this slide up, it makes me think of Luke Skywalker. But Pasteur was, one of the key people to link medicine and veterinary medicine. You could see the links between human health and animal health, in particular domestic and agriculture environments. And Virchow, this guy is, he's one of my heroes. He's, the father of modern pathology. This guy, you know, described thromboembolism. He described, you know, work around the social links between infectious diseases and social science in relation to things like typhus. He was ahead of the game in terms of things like meat inspection, in terms of trichinella. But one of these things that he said was, between animal and human medicine, there are no dividing lines, nor should there be. And that absolutely applies when we're talking about antimicrobial usage, antimicrobial resistance. And then we fast forward probably to Calvin Schwab, who's really kind of, he's the one who's really sort of given the credit for coining the phrase one medicine. So this link then between human medicine and veterinary medicine. So Calvin had a joint appointment between the Human Medical School and the School of Veterinary Medicine at the University of California, Davis, and produced this, you know, kind of his seminal text, you know, Veterinary Medicine and Human Health. So this kind of concept of One Health, how is it distinct from veterinary public health? Well, it goes way beyond that. You know, but it's very much then about the collaboration that we get between disciplines. And one of the things that kind of comes out here is not just what we can learn between human medicine and veterinary medicine, but how we can then take that to the next level and actually be involved in much more active collaboration across the life sciences sector. I still quite like this kind of little cartoon that came from Sweden, and obviously in relation to in relation to where we are and what we're talking about, you know, you can see here antimicrobial resistance sort of sitting alongside bacterial infections, viral infections, vector-borne infections, under this umbrella alongside zoonotic infections. And there's, again, you know, I think that's quite useful in helping us to think about what One Health is beyond this kind of fluffy paradigm of people, animals, and the environment. It is all of these things, and there are opportunities here for us to learn from one another. So I'm delighted, you know, that there are people here today from pharmacy, there are people here from human life sciences, there are people here from human medical background, there are people here from animal health and agri-food and biological sciences, so a whole range of different disciplines. And we're seeing more and more of the social science then coming into that as well, and the environmental aspects then linked to that too. There seems to have been a bit of a trend within the sort of One Health movement to define and redefine what One Health actually means, but this is probably one of the most current definitions. So One Health is a collaborative, multi-sectoral, transdisciplinary approach working at local, national, regional, national and global levels to achieve optimal health and well-being outcomes, recognising the interconnections between people, animals, plants and their shared environment. So I think we can probably all take the bottom bit as read. People, animals, plants and their shared environment. Okay, plants snuck in there. We can think of it in terms of all those different levels. So it's not just this kind of ethereal, sort of 10 years ago, it was sort of Chatham House and people like that. You know, we're talking about One Health on a sort of global level. But we can take that right down to local and even regional level now. Okay. Optimal health and well-being outcomes. Again, we talk about One Health, but actually we probably, if we were going back to recoin the phrase, we would probably talk about One Well-being, because I think there is an aspect related to health and welfare within that. However, the most important part of this definition is in the first line. It's the collaborative, multi-sectoral, transdisciplinary approach. And that's why you guys are all here today. Is to have those discussions in and around how we can work across different disciplines and use our relevant expertise. Dame Sally has been on a journey. Whenever she was Chief Medical Officer for England, she was quite outspoken early on about how vets and farmers were using bucket chemistry, you know, in terms of their usage of antibiotics. But you can see here that, you know, a more recent quote, she said, "In the UK, we believe that the major driver of resistance is prescribing for humans. Nevertheless, we appreciate that good farm management, biosecurity and animal husbandry systems are vital to minimise the occurrence of disease and therefore reduce the need for antibiotic use." So there is a realization there now, and Dame Sally was one of those people who's been on that journey, that through a One Health approach, through that communication, through that collaboration, she has realized, that it's not just all about the vets and the farmers using antibiotics by the bucket load. But there actually is a piece there to be worked out. And, I mentioned the social science, but how we change behaviours towards antibiotics and change attitudes towards how we use antibiotics is really important. So I'm not going to jump into a whole pile of detail. This is just a sort of introductory kind of, to kick things off. But for as long as we've known about how antibiotics work, we've also started to understand more and more about how resistance works. Okay, so while there is that, even going back to, Fleming and his acceptance speech for his Nobel Prize around the use of penicillin, already he was talking about the possibilities or the likelihood of resistance. And let's face it, a lot of the bacteria, a lot of the antibiotics we use were derived from soil bacteria. And a lot of those soil bacteria also derived antibiotic resistance factors as well. So that tension has been going on for millennia, even before we discovered antibiotics, let alone antibiotic resistance. But how we use antibiotics is our responsibility. And that is something we need to be quite careful with. I've been involved in establishing an organisation called Vet Sustain, and I guess really what I wanted to say here is, in terms of overall sustainability, again, it's another one of those sort of buzzwords that's out there in the minute. When we think about sustainability, antibiotic usage is absolutely part of that. Okay, how we use antibiotics, how we use anthelmintics as well in relation to farm animals is critically important. But these are our 6 veterinary sustainability goals: so diverse and abundant wildlife, net zero warming, a no waste society, a good life for animals. So this is up-to-date animal welfare thinking. Health and happiness, that's our animals, it's our farmers, it's our pet owners, it's our veterinary surgeons as well, and enough clean water for all. So you can see there within that there is a clear line for including responsible antimicrobial usage within that kind of concept as well. Lots and lots of reports. This is an area where there are huge numbers of reports kind of coming through. And I've just picked out a few of these. Obviously, O'Neill's report, was quite a sort of seminal one in terms of One Health crossing over. So that's the 3rd one across. Looking at both human and veterinary medicine and tackling drug-resistant infections globally, and some of the recommendations coming from that. We have been really fortunate, I think, that there's been reasonable kind of join up over this last decade. We're now into the 2nd 5-year National Action Plan, around tackling antimicrobial resistance. And these are cross-Whitehall activities now. So we're seeing much more of a join up between the Chief Medical Officer, Chief Veterinary Officer, and pharmacists and policymakers across central government. And we're also fortunate to have seen that here in Northern Ireland as well, where our CMO and CVO in Northern Ireland have been working very, very closely in and around AMR as well. And we would hope that we would be able to see that going forwards too. But in particular, I want to draw your attention to these 2 reports. If you haven't seen them, these are both very recent. I had the opportunity at the start of September to travel with Innovate UK on a global expert mission on AMR to the United States. And even though I've been sort of advocating for One Health approaches for the last few years since I was president of BVA, and getting that sort of joined up approach going, I got my eyes opened on that mission as to where some of the big challenges are in human medicine, in and around things like antifungal resistance, treatment of patients under cancer chemotherapy, cystic fibrosis patients. So real challenges. And certainly, on that mission, having the opportunity to talk with some of the human life sciences people about the challenges facing us in veterinary medicine was really interesting as well. They were absolutely astounded that we didn't really have any functional antifungal, licensed antifungal agents for food-producing animals, for example. But I would draw your attention to these two reports. The first one came out sort of early October, tackling antimicrobial use and resistance in food-producing animals, lessons learned in the United Kingdom. So this is a UN FAO report, and it's showcasing some of where we have done some really good stuff here in the UK around food-producing animals. And just within the last week or two, the latest report has come out from Veterinary Medicines Directorate. It's always a year out of date because it's, historical data. But the VARS report, talking about veterinary antibiotic resistance and sales surveillance report, we're now down to 28 mg per kg, okay? That's amazing, okay? We've made a 60–70% reduction in our antibiotics usage in farm animals since 2014. And certainly for HPCIAs, we're now down to about 0.12 mg per kg, which is, again, just absolutely phenomenal. There are some little blips. So I'm an aquaculture vet, and, salmon have seen a slight increase over this last couple of years. But once you get down to pretty low levels, you're going to sometimes see a little bit of that as well. So look, I'm going to leave you with this. Lord O'Neill said, although AMR is a massive challenge, it is one I believe is well within our ability to tackle effectively. Thank you for joining us today. Hope you take the opportunity to have lots of chats today, swap business cards, swap email addresses, and take the opportunity then to make the most out of the networking today. Thanks very much.
How much taller he is than me. Thank you very much, Simon, for that. We are going to leave questions for Simon to later on because he's here with us all the time. Now we're going to move on to the other younger people among us, the younger researchers who've been doing some work on AMR. Before we continue, can I just say, please put your phone on silent because most of this is being recorded and will be shared to people who couldn't make it here today. So if you can put your phone on silent, if you need the bathroom, the toilets are just behind you, but at the bottom of the stairs over there. So our next speaker is Aoife Rogers, and Aoife will be telling us all about her work she's doing on gut pathogens and microneedles. Thank you.
Good morning, everyone. I'd firstly like to thank the organisers for giving me the opportunity to talk this morning. I'm a postdoc researcher in the lab of Professor Ryan Donnelly in the School of Pharmacy. And this morning I'm going to talk about some of the work we've been doing looking at the effects of antibiotics on the gut microbiome and the potential for microneedles arrays to circumvent gut microbial dysbiosis. So over the last number of decades, it's really become apparent that the gut microbiome plays a really important role in maintaining health and homeostasis. The problem comes when this imbalance is disrupted in a process referred to as dysbiosis, and this can occur due to a number of different reasons, such as aging or comorbidities. But antibiotics are the main inducers of gut microbial dysbiosis. And this has a number of implications for the host, including infections such as C. diff, which can go on to cause colitis. But it also has implications in terms of antimicrobial resistance because the gut microbiome contains important opportunistic pathogens, including members of the escape group, which can subsequently take over. Now, there had been a number of studies that had suggested that the effect of antibiotic delivery route could affect the degree of dysbiosis, but these results were quite contradictory, and we wanted to explore this further. To do this, we selected two commonly used antibiotics that are delivered both orally and intravenously, amoxicillin and levofloxacin, and we delivered these in rats, both orally and intravenously, and we subsequently collected the fecal pellets and carried out 16S sequencing. And this essentially shows that with both amoxicillin and levofloxacin, we do see differences in dysbiosis compared to oral and IV delivery, but the effect with amoxicillin was much more rapid in comparison to that with levofloxacin. We subsequently carried out taxonomic analysis, and essentially what this shows is that both antibiotics induce different levels of changes in the gut microbiome and the families that are actually altered. And importantly, both intravenous and oral route cause different levels of dysbiosis in the gut. We subsequently looked at the longer-term effects of this, and essentially what we showed was that with amoxicillin, when you deliver antibiotics intravenously, the level of dysbiosis seems to return to baseline much more quicker in comparison to antibiotics delivered orally. However, we don't see the same effect with levofloxacin, showing that it's antibiotic-specific. This would imply that conventional parenteral delivery systems may play a role in circumventing this gut microbial dysbiosis. In this regard, we were interested in looking at microneedle arrays as a means of bypassing the gut microbiome. And essentially what microneedle arrays are they are essentially a series of microscopic needles attached to a base support. And once inserted into the skin, they can deliver drugs quite easily. They're usually fabricated such that they're long enough to bypass the stratum corneum, but they're short and narrow to avoid stimulation of dermal nerves, and in this way they allow a really simplified method of delivering drugs such as antibiotics that patients can actually deliver these themselves. And these are just a few images of the different types of microneedles available. Depending on how the microneedles are fabricated, they can deliver drugs in different ways, and there's mainly 5 different groups, including solid, hollow, coated, dissolving, and hydrogel. And for the purpose of this talk, I'm going to talk specifically about hydrogel-forming microneedle arrays. So these are microneedle arrays that are fabricated from a swelling polymeric material, and they're used in conjunction with a reservoir containing the drug, such as the antibiotic, for example, a directly compressed tablet. And they're inserted into the skin in their solid form, and once inserted into the skin, they begin to take up skin interstitial fluid and begin to swell, and then they subsequently pull down the drug that's contained in the reservoir on the surface of the skin. And in this way, it's a really painless and simplified method of delivering drugs such as antibiotics. So we were interested in using these to deliver both amoxicillin and levofloxacin as a potential means of bypassing the gut microbiome. And to test the feasibility of actually delivering antibiotics by this route, we carried out pharmacokinetic studies in rats. And this is just a schematic of the process we use to apply the microneedles. So they're essentially used in conjunction with a directly compressed tablet that's used to apply on top of the microneedle, and this is held in place with a foam adhesive border. The rats are subsequently shaved, and then the microneedles are applied. We either apply 2 or 4 patches depending on the dose that we want to deliver. We subsequently carried out pharmacokinetic studies to see if we could essentially deliver these antibiotics, and this is the pharmacokinetic studies for amoxicillin on the top and levofloxacin on the bottom, and we compared this to oral gavage. And as you can see, over the time period, over 24 hours, we could successfully deliver both amoxicillin and levofloxacin. And studies are currently ongoing to test whether the microneedle arrays can circumvent this gut dysbiosis that we see with IV administration. And with that, I'd like to thank the Donnelly Research Group and Wellcome Trust for funding this project, and I'm happy to take any questions.
That's really, really interesting work. I've always been fascinated by the microneedle business. So we have a time for a couple of questions for Aoife, if anybody wants to ask Aoife something. Anyone? Yes.
Do you think you're going to see a difference in the absorption or the administration of different classes of antibiotics?
Yeah. So that's a great question. And I think that's probably the reason why we see differences in terms of the dysbiosis with the amoxicillin and the levofloxacin. So the oral bioavailability between the 2 antibiotics is drastically different. For example, amoxicillin you see about 90%, whereas levofloxacin is much lower. So that's probably why we're not seeing the difference when we compare the IV to oral administration, the dysbiosis that we see. Whether we see differences with the microneedles or not, we don't know yet. Those experiments are currently undergoing.
Any final questions for Aoife?
Yes.
Thanks, Aoife. you said a tablet on top of the microneedles rather than incorporating the antibiotics directly inside the material. Is that just because it's more convenient to do that, or can you not really give needles up enough?
So there's a couple of different approaches that we can use to deliver different classes of antibiotics. And that's a great question. For the example, for these drugs, we use the hydrogel form in microneedles because they allow for sustained release over time. We have, for the likes of aminoglycosides, such as gentamicin, we've used dissolve in microneedles because you want, depending on the pharmacokinetic profile that you want, for example, gentamicin, you want peak and trough plasma levels because sustained release is associated with toxicity. Whereas with amoxicillin, you do want to see that sustained release. So we sort of modify the microneedles according to the drug that we're wanting to deliver. And for this purpose, we use the hydrogel form of microneedles. So yes, they don't contain any drug themselves. It's just the reservoir that's contained on the surface of the microneedle. And then once they start to swell, the drug is pulled down through the reservoir. But we have done studies with the likes of gentamicin where we've used dissolve in microneedles and looked at the pharmacokinetic profile of those. And we do see that we can modify the pharmacokinetics based on the dissolution rate of the microneedles. But that's a great question, and it does depend on the class of antibiotic and the pharmacokinetic profile that you want to see. So yes.
Thank you very much. So let's just thank Aoife again for that wonderful talk. I think most of our speakers are going to hang around after this, so please, if you have more questions, do go and ask them. And our next speaker is Dr. Tony Benson, and he will be telling us about factors influencing responsible antimicrobial usage on farms.
Hi everyone, my name is Tony Benson and I work here in IGFS. I work with Professor Moira Dean, and our group really looks at the psychology and the factors underlying behaviour. So following on from what Simon's saying, we're taking a kind of social science approach, looking at AMR and antimicrobial usage. So in terms of antimicrobial usage and antimicrobial resistance, we have been involved in a number of different projects. There's been a few PhD studentships, and more recently we were involved in a safe food project. So as part of the safe food project, we conducted a survey with farmers. The project also asked vets about their antimicrobial usage and their kind of behaviours around antimicrobial usage. But just in terms of timing today, I'm just going to focus in on the farmers survey. So as many of you here will know, imprudent antimicrobial usage, or AMU, accelerates the development of antimicrobial resistance, AMR, diminishing treatment efficiency and endangering the future of animal and human medical treatment. And international reports have recognised that AMR is a global health emergency with serious health implications such as prolonged illnesses, fatalities and also economic impacts. And in response to AMR concerns, there have been global frameworks developed, such as the World Health Organization's Global Action Plan and the European Parliament's One Health Action Plan. And in summary, basically, these reports recognise that there is a need for a multidisciplinary and a holistic approach across organisations and countries across the globe to tackle imprudent AMU. And as Simon said, this importance of collaboration and coming together. So in addition, the use of antimicrobials in agriculture has also been recognised and the importance there. And the European Parliament has drafted, and they brought in legislation which came in January 2022, prohibiting prophylactic antimicrobial use across in agriculture. So this project really aimed to obtain a better understanding of the knowledge, understanding and attitudes of key stakeholders, so farmers and vets, so I'm just going to focus on farmers today, to identify factors influencing decision-making and current antimicrobial usage behaviours, and this was an Island of Ireland study, North and South. So for our study, we based our study on the COM-B model. So it's kind of like a psychological model which says that in order for a behaviour to occur, there has to be three things there. There has to be capability, there has to be opportunity, and motivation. So in terms of capability, capability is basically an individual's physical and psychological capacity to engage in a behaviour. Opportunity is all the things outside of the individual which kind of prompt or promote a behaviour. So this can be things such as the physical infrastructure or social relationships. And then motivation is all the kind of brain processes that drive and energize behaviour. So these can be reflective motivations, so where you think about a behaviour and you think about the pros and cons before you act, or it could be more reflective, more automatic reflections, such as habits and our emotions, which can also motivate our behaviour. So we wanted to use this model to basically understand farmers' responsible antimicrobial usage. So I don't really have time, but basically just this slide says that we did go for farmers across a range of different sectors, and we aimed for a spread of farmers. We ended up with a final sample size of 392 farmers. So in terms of the results, with regards to, we looked at knowledge, so objective knowledge and subjective knowledge. So for objective knowledge, we had 7 statements which you asked the farmers to say whether they were true or false. We also asked them about their confidence in the answer that they gave. So rather than going into the score in detail, basically the farmers could score between 0 and 35, and the participants, the farmers in this study, had a relatively high objective knowledge, so they had a mean score of 25.55 in a range out of possible 35. But we also looked at subjective knowledge. So objective knowledge is what you know, and subjective knowledge is what you think you know, and they're two different things, and they can be quite important differences there. So in terms of subjective knowledge, participants rated their self-perceived knowledge in 4 statements such as these. And overall, participants believed they had a relatively good knowledge of responsible antimicrobial usage, with a mean score of 15.83, ranging from 4 to 20. And we also asked the farmers about their awareness. So 80% declared that they were aware of the consequences of AMR, and 70% were aware of the concept of One Health. So before I go into the results kind of based around the COM-B model, I did want to say that our kind of output variable, or the thing that we were concentrating on, was responsible AMU behaviour. So antimicrobial usage is important in agriculture. It doesn't need eliminated. It's there. Antimicrobials need to be used. They're important for things like animal welfare. But what we want to do is promote responsible AMU behaviour and prudent AMU behaviour. So when we put the 3 parts of the COM-B model, so capability, opportunity and motivation, into our model, we found that they explain 70% of the variance in responsible antimicrobial use behaviour. And we also included socio-demographic characteristics and things such as farm characteristics like length of or size of the farm and length of time on the farm. So the sociographic factors predicted approximately 5% of the variance in responsible antimicrobial usage behaviour. Capability then above that predicted approximately 3% of responsible AMU behaviour, motivation 8%, and then opportunity had no impact. So within the kind of COM-B model, if you go down a little bit further in terms of more specific factors and individual variables, the number of years experience, emotions and subjective knowledge were all very important predictors of responsible AMU behaviour. I'm just going to focus on these each in turn. So in terms of years experience, so more years experience correlated with more responsible behaviour. And this was coupled together with the finding that farmers with less experience depend more on peer and social support than those with more experience. So basically to improve experience and then hopefully improve responsible AMU behaviour, farm visits or discussion groups may prove useful, where farmers come together and they model good AMU behaviour, and they kind of talk about good antimicrobial stewardship strategies that they have in place. In terms of subjective knowledge, increased subjective knowledge increased with responsible behaviour, and this pairs with findings from the literature previously, which found that knowledge is a strong predictor of responsible AMU behaviour. So to improve knowledge and hopefully improve responsible AMU behaviour, continuous education channels such as online training materials or webinars or interactive platforms for farmers might be useful. And in terms of emotions, the farmers were asked, if you had to stop blanket use of antibiotics, how would you feel? So feeling satisfied, wise and calm, all correlated with better practices. So to try and instill these emotions, facilitated discussion with other farmers that have had a positive experience to support and empower them to make changes on the farm could be useful. And just to summarise, so farmers are interested in reducing antimicrobials, and there's something that they're aware of and that they want to do. Strategy design should encourage incremental behaviour change so the farmers and vets feel capable to implement antimicrobial stewardship strategies. Behaviour change theory should be included in strategy design to address barriers experienced in relation to AMU in agriculture, and overall careful consideration and an evidence-based approach are required to develop intricate interventions and strategies for successful and sustained behaviour change. So I was only really very loosely involved with this project. Moira was the PI and Claire was the main postdoc with Sarah, the PhD student. And in kind of recognition with this, rather than taking questions, maybe better directing these to Moira. She said that she's happy to take any questions on the project or anything to do with our work if you want to get in touch.
Thank you very much, Tony. I think you have carefully dodged questions there. So I'm sure that people are going to still catch you afterwards anyway and have a chat about this. So we are going to move on to the next speaker. Can I just ask that if you're taking photos for Twitter or anything like that, just don't for this next speaker, please, or videos or anything like that. So our next speaker is Lucy Dillon, and Lucy will be telling us about machine learning models used to reveal the key role of accessory genes in defining AMR phenotypes. Thanks, Lucy.
Yes. Okay. Hi, everyone. I'm Lucy Dillon. I'm a third-year PhD student in Parliament.
Thank you very much, Lucy. So I think we've come to the point where I think a lot of us have been waiting for. So our next speaker is our guest speaker that has come all the way from Liverpool to join us today. So thank you for coming to listen to him. And that is Dr. Adam P. Roberts. I have a little summary of who he is that I can read out to you. So Adam did his undergraduate degree in applied biology at the University of Coventry. He then went on to do a molecular microbiology PhD at the University of London. Then he spent around 20 years in University College London as a postdoc, a lecturer, and a senior lecturer. It doesn't look it, I know, 20 years is a long time. And then in 2017, he moved on to Liverpool School of Tropical Medicine, where he's been working ever since. And prior to that, he started this wonderful outreach program, Swab and Send, which I'm sure some of you would have come across either on Twitter or something like that. If you haven't, please go look it up, and Adam will be telling us about that today. Adam is also an advisor on the Longitude Prize, if anyone knows what that is. So that is a prize that helps to determine what the focus should be, and the Longitude Prize is currently focused on AMR. And he's also an advisor on the Royal Society of Hygiene and Tropical Medicine. He has, as I said, pioneered various outreach programs, including, I think Swab and Send get samples from all over the world, and includes school pupils, the public, and so on. He coordinates an antimicrobial resistance research network funded by the European Union and includes European and African researchers called JPI-AMR. So you might want to be part of that network. And he wanted me to tell you a fun fact about himself, that in fact, he's an accomplished triangle player. For those of us who don't know what that means, I didn't know. It means that he can only play one note. So that means he has no inclination at all. So I would like to welcome Adam up to the stage as he gives us his talk today.
Thank you very much, Linda. That's a marvellous introduction. I do play the triangle very well, by the way, and I can demonstrate it if anybody has a triangle with them. It's an absolute pleasure to be here. It's my first time in Belfast. It's my first time in Northern Ireland, and it's going to be really enjoyable for me to sort of reflect on the experiences I've had with one of my projects over the last 7 years and highlight some of the mistakes I've made. So hopefully, people in this room who will be involved in Linda's outreach project won't make the same mistakes. Okay, so the title is, How Can We Engage Citizen Scientists in Our Fight Against AMR? Insights from Swab and Send. And I've put these eyes here, and they are kind of a reflective note from me when I was going through my talk over the last few weeks about things I can impart, and if one of those makes a difference to your own citizen science project, then I think it may be useful. Okay? I might not remember what they all are, but we'll see when we come to them. Okay, so as Linda said, in 2015, I started the Swab and Send Citizen Science Drug Discovery Project, and I primarily started that to avoid things like this sort of screenshot here. Is this a laser as well? No. This screenshot here, which just illustrates the lack of understanding sometimes that becomes apparent with the general public and other sectors of society. So I think this was a discussion on a new type of vancomycin, and somebody stated that, you know, that won't be resistant to the bacteria. That level of understanding is really difficult to try and tackle. You've really got to go deep in education to try and address that. Citizen science is quite ingrained in antibiotic drug discovery. The original Penicillium chrysogenum strain that is still used to produce penicillin around the world was actually the second strain. So the first strain was Penicillium notatum that Alexander Fleming found. And it wasn't a very good producer. It's pretty rubbish, actually. So they had a citizen science project over in the States calling for moldy things to be sent into a lab. And there was a moldy cantaloupe sent in by somebody who worked in the lab, and it had the Penicillium chrysogenum strain on it. And I think it produced something like 2 orders of magnitude more penicillin than the original strain. And another reason I did it was just to increase awareness of AMR and the issues surrounding the difficulties in both finding and developing antimicrobials, in order that people would value them more and then take better care of them and not sort of be offhand about using them or sharing them with family members, which is some top-down advice we got recently. And why did I do it? Well, I thought societal understanding was one of the prerequisites of a solution for management of AMR. This was highlighted in the O'Neill report, which has been previously mentioned, but I think we can all agree that if we can tell people about how important antibiotics are and how they underpin our society in various sectors, whether that's healthcare, food security, etc., people will begin to understand that constant and sustained message and look after them better. And that's one of the insights that I would like to share. Consistent messaging with a citizen science project is important. Always have the same message that you reiterate over and over again. Okay? And also have a citable source that people can go and get more information from. So it doesn't really matter what it is. It doesn't need to be in a high-impact journal. It could be in a new journal, for example. Access Microbiology from the Microbiology Society is a very good format for an editorial describing a new citizen science project which you think is going to be sustained. This just gives people the opportunity to go and read it in their own time. There's mine on Swab and Send. Please have a look. Make it open access as well. So, little overview about what we do with Swab and Send. People hear about it, usually on social media. So we've got quite a big Twitter presence. However long Twitter lasts for, we might have to move on to Mastodon or whatever it is now. But we also put all of the data from the project on Facebook. And I'll come back to that, why we use Facebook, in a moment. So marketing your project on Twitter and other social media platforms is really important. Okay, you have to make all the mistakes to learn which sort of strategies work. I've done them all. So people in my project, the Swab and Send, they pledge a small amount of money to take part. It's pay and play. And the reason is, really early on, I had no funds for this. It was done after hours. It was done by myself and postdocs who were enthusiastic about the project. And we had to cover the minimal expenses that it took to run it in the lab. And people seem quite enthusiastic to pay. We've got thousands of pounds worth of pledges from this project. And it also kind of acted as a control as well, because what we found out is if we give swabs out for free, there's a lack of ownership and we very rarely get them back. So it's just a waste of resource. We've given out 100 swabs at a particular event at University College London in 2016. So we thought there'd be quite a lot of enthusiasm. You know, it's a UCL project originally, and we got one back. And that really changed our minds, actually. We're not doing this again because it's just a waste. You know, 100 swabs costs over 100 quid. So we're down on the finances. But after moving to LSTM, they made a nice, easy online donation portal. And you can get tax relief as well, so the school gets a bit more money. So you can do it right, and it can cover the expenses needed. So we send out the swabs. We send them out in envelopes to anywhere in the world. There are other discussions we can have about that and the Nagoya Protocol for this particular project, about receiving swabs from international sources. Most of ours are from the UK, which makes things a lot easier. People then swab whatever they want. This is a hissing cockroach at the New Scientist Live exhibition in 2018. We get swabs from pretty much anything, anyone, and anywhere you can imagine. Some of the things we get are awful. Some of the things we get are fantastic. So I'll give you an example. If you receive a swab and you own a cat, there seems to be an overriding impetus to put the swab in the cat in one of the multiple orifices. We don't know why, but we have an overrepresentation of cat orifice samples within our library. It's a weird thing, but we're quite agnostic to the samples that we take. And we're agnostic to the samples that we take because what we want to do is de-replicate finding the same bugs producing the same antibiotics that we would pick up if we just sampled soil swabs. So we essentially want to swab the rest of the planet. Everything that hasn't been swabbed yet, we're quite interested in. And then we can't really turn our nose up at cat swabs. But anything you can imagine, we've probably got a swab from it. And we're always interested in new things. So anybody got any ideas, let me know. I will send you a swab for free based on your enthusiasm. The swabs are then returned. These are all from Northern Ireland, from the British Medical Association event a couple of years ago, somewhere over here. I can't remember. There were hundreds returned, which was quite nice. And then we do the microbiology on them. So we plate out the swabs onto brain heart infusion agar. It's nice and rich. It's got 4% agar in it to stop the swarming bacteria overtaking the plates. These are all things that we developed. And as you can see here, we get quite a nice array of diversity most of the time. Okay, so that's really nice as well. And then this little pair of eyes here is just to kind of highlight the fact that pictures are fantastic during your work. So Linda, take pictures of everything you do, every stage. You never know when you'll need a good picture. We also do events and campaigns. So this is us down at the New Scientist Live. So you can see here how many of us were in the team. So I had to cover all of the accommodation costs for that. That cost me about £5,000. It involved two different grants supported by the Microbiology Society, I would say, which is marvellous. And we also had gimmicks. Okay, we use our imagination to incite interest in the public. So you can see over there on your right, the Tower of Swab, which was a 12-foot high swab with other swabs sticking out in a double helical structure, which was marvellous. So we essentially sampled the entire aerobiome of the conference over a 5-day period. And it's amazing how many people will come up and have a look and then touch the swabs, and we even saw one person putting them in their mouth. You can't really explain that, but great. And we got lots of diversity, which is what we're after, okay? And nobody had ever done that before. So there's a nice little Twitter post, and oh, maybe I'll have a go. There's £30. And it just sort of becomes self-sustaining, which is nice. A little bit sort of closer to home, my building, you can just be seen there, that's the Liverpool Life Sciences Accelerator. This is one of our car parks, and underneath it is one of the last sort of sites of the Victorian slums in Liverpool, and the local archaeologist team came and dug it up for a survey. So obviously we went down there and jumped in, said hello, and swabbed everything. And because of that enthusiasm on our part to go and mess around in the mud and the shoes and the bits that they pulled out of the ground, we now get yearly invites to their archaeological digs all around the northwest, which is great. We were just down on the pier head a few months ago, where the team was, looking at some old things on the dock that they'd just uncovered. It's fantastic. This is down at the Science Museum on the lower part of the slide. I think we entertained about 300 people there at a New Scientist Live late exhibition. So we were one of about 6 or 7 different teams. That was sponsored by UKRI. So if you do these things, you can also claim costs. I think we managed to recover about £3,000 for that, which is great because it helps pay for all the thing. Just as an interesting thing here, those agar plates are actually full of jelly, so they're edible. So this really interested everybody when the team just started eating them. So, you know, do you want one as well? They were like, you microbiologists are crazy people, which is quite nice. I've highlighted agility here. Obviously, we had lockdown. Nobody could go out. Nobody could do any swabbing. So the project was at risk. So what I came up with was the fact that we could all make Winogradsky columns. We could all go out with an empty bottle, get some dirt, put some water in it, etc., and then see what grows. And then when we come out of lockdown, we then swab it, send it over to my lab. We'll have a look what's growing. We've actually got some hits from there. Not these are mine. some Winogrossi column samples came over from America, and we've got some antibiotic producers which can kill E. coli really, really well. We're quite excited by those. So again, that sustained activity, it means that you've always got a presence on your social media platforms and you're always doing something within the project, so interest doesn't die off. Be proactive and fun. So this is kind of something that we did in-house a few months ago. These are some of the samples that receive. Okay, so there's a kind of a warning here. Be wary what you wish for. So we like getting the swabs back in from all over the world. Sometimes people send in different things. So we had holy water sent in from somebody whose relative had just died. Actually got some interesting stuff from the holy water, and there's a whole host of social things that we could do there on various media platforms, but we want to be respectful, so we won't. That was something, I think that was a cat skull from one of the archaeological digs. And then by being proactive, I mean, you know, put yourself forward for awards and stuff. We won an award from BioNow, an industry sort of group within the Northwest earlier this year. So the first thing we did was swab the award. You know, again, nice social media. It makes interest sustained. And also linking with synergistic activities. So this was one of my labs earlier this week. We've got a public health campaign going on, sort of linked loosely with Swab and Send. Some of the, I can't remember which public health institution it is, but they want to basically do a campaign to encourage people to start washing their hands again, because that's dropped off. Now COVID is a thing in history and doesn't exist anymore. So one of the labs is turned over to that. And all of those bacteria that we get from their swabs, they go into Swab and Send and into the drug discovery. So it just increases the amount of isolates that we are doing. Now there's a warning here as well. Okay, so I've put red eyes for danger. And this is about protecting your own work. So if you start to generate success in a citizen science public engagement project, it can very quickly take over your entire time. Your time is a resource which is limited. I could have spent in previous years all of my time on Swab and Send, and that is to the detriment of your more traditional scientific research. So you have to find that balance. That balance might be because you're including more people, or actually you start saying no to things. Okay, it's an important thing to consider. So what do we do with the microbiology? So we've got these plates from swabs from all around the world. We plate it onto BHI. We've also toyed with different agars. So for example, Sabarad here, chicken gravy agar. So I noticed that the ingredients of chicken gravy were very similar to brain heart infusion. So we tried it, and you can see over there on your right, it works really well. This has actually got real world implications for low resource settings, whereas a bottle of brain heart infusion agar would cost about £120. Chicken gravy agar is about £1. So you can actually carry out reasonably good microbiology in resource-limited settings using a different type of media. This is an example of the kind of return we get on the brain heart infusion plates. This was from the Institute for Microbiology and Infection at University of Birmingham. We did their summer school microbiology component, and you can see, get lots of different things, and you can see that there's lots of different things sampled. And that was really good, and again, that was funded as well, so we got a financial return. So each one of those morphologically distinct colonies is picked into a well of a microtiter plate, and we go for redundancy over exclusion. So if we're not sure that two different colonies look the same, we take both of them. Like if there's 5 or 6, we take all of them. We don't mind isolating the same thing more than once. We just don't want to miss anything. Those are then replica plated onto various agar plates using the 96-pin replicator, or the hedgehog as we call it. So you can see here one of the examples of an agar plate with nothing else on it, no other indicator strains on the background. So they're all gridded out in nice format. We know where they've come from, we know which swab they came from and what was swabbed. And then you can see here, this is kind of one of our indicator strains. Micrococcus luteus has been plated out onto the background prior to stamping with the 96-pin replicator. And you can see here and here these nice zones of inhibition. Okay? Micrococcus luteus, for our purposes, is useless in terms of drug discovery because it's non-pathogenic, but it is yellow and it's a real weakling. So you get nice zones of inhibition. And that means that people who participate get really pleased. It's like, okay, I found a bacteria from my teddy's nose which can produce antibiotics and kill this bacteria. Let's get the rest of the family involved. Let's make another donation. Let's do the project again and again. And we get that quite often. But what we're really interested in is activity against the various pathogens. And currently we're looking at E. coli, Candida albicans, Candida auris, MRSA. We have also been looking at Pseudomonas aeruginosa. We are probably going to be looking at TB soon as well, because we've built up quite a resource, which I'll come to in a moment. So that red flashes, you can see a nice zone of inhibition against E. coli, and that's really what we're looking for. Now, at this particular stage in the project, that ends the contract that we have with the participants. Okay? That's as far as we are going to tell them at this time. Because once we've found some phenotypic activity, then it goes into the more traditional drug discovery science, and we can't really reveal what we're finding, because the best way at the moment to kill an antibiotic discovery and development project is to tell people about it without any IP. That may change in the future, we'll see. All of the results to that point are put on our Facebook project page. So an insight here, make the public interface as easy and as cheap and accessible as possible on both sides. So it has to be the minimum work for you running the project, and it has to be the minimum number of clicks to find the data for the participants. Facebook is free. You don't actually need a Facebook account to view this page, which is quite nice. There's only been minimal kickback with the whole anti-Mark Zuckerberg thing, but we can cope with that. Okay? Looking for perfection is probably going to be inhibitory for progress of your project. Something is better than nothing. Okay? So, what do we do with all of these things? So this insight and legacy, traditional scientific discovery. Okay, so that's the end of the citizen science bit, but what happens then? Okay, it's citizen science, so it's got to somehow go into normal scientific activity within your research group. So we would take one of these, okay, and we would put it in a new microtiter plate, and that would be a species-specific hit plate. So for example, with the E. coli's, we've probably got over 100 hits now. So these are bacteria from many different places which are able to inhibit or kill E. coli. We then plate those out again, because what we've noticed is that actually we can get loss of activity depending on where they're growing on those plates. So the neighbors influence exactly what they're producing. It changes gene expression, etc. But to make things simple for us, we want things that come through both of these assays. And then we go into cell-free supernatants, okay? Because we want to know what it is that the bacteria are producing. So within the agar plate assays, it looks like whatever is being produced is diffusing into the agar because we get the zones, but we want to make sure it's also in broth, because then we can work on it. So we do the cell-free supernatant assays, we grow it up, either at 37 or at room temperature, depending on where the isolate came from. And then we can run over 24 hours growth curve analysis with our indicator strains. And when we get hits there, we can then proceed to fractionation. And that was a key sort of bottleneck in our process. And it's all about process. It's all about moving those rate-limited steps all the way to the end of the process so you can do everything you want rapidly. And we'll come back to the fractionation in a minute. So, also, along the way, we find many interesting things, far more things that we can work on. So you've got here, for example, interesting zones of inhibition. You've got interesting colony morphologies. So that's the microbiology from the cell-free supernatant assays. What we also do is sequence the genomes of all these. That's really useful, actually, because we can drop them quickly. We want to fail fast on a particular isolate. We don't want to work on it if it's already known. So if we sequence the genome and we know what it is and we know it's likely producing an antimicrobial, which is already known and published, we forget about it. But what there might be is a nice story in there which we can take for a lab project, and that benefits everybody in the group, usually with a minimal amount of work. So by sequencing it, we get strain identification, we can carry out comparative genomics, we can identify the biosynthetic gene clusters, and we can have spin-off projects. So for example, this one we published a few years ago, a Klebsiella gramontii isolate on a reusable water bottle. And I'll highlight this one because, again, there's a word of caution. So this particular isolate came from a swab from the Telegraph newspaper offices, and they wanted to do a little bit of work with us based on swab and send. And I said to them, I said, we'll do the work, but we want you to treat microbiology sympathetically. We don't want any shock stories. Oh, there's nasty things, you're all going to die. And they agreed, but they agreed in a strange way, and it sort of got the hackles on the back of my neck rising. Anyway, they came out with their piece, and it was all great. And then the following day, a different journalist came out with another piece in their health supplement saying, you know, this alarmist, Dr. Roberts, going on about, because this is quite a pathogenic strain, it was on their water bottles. I said, you know, you may want to clean your water bottle as you would a cup at home. Why are you not doing that? I asked them to scrape the end if there was a biofilm on it, and it was. And we showed it was a biofilm former. So that really irritated me, and you never really irritate a microbiologist when they've got bugs from you and your samples. So we sequenced it, we showed it was pathogenic, and we published it, and we said it came from the Telegraph offices. So there's a little bit of a hate publication there. But anyway, it's great for generating interest in your project. So again, it's the sustained interest, which is important. Now, going back to our hit plate, this is kind of what we do. So we do the cell-free supernatants, and we do this in order to prioritize which ones we're going to work on, which ones we're going to sequence, and take forward for fractionation. So this is the kind of graph that we get from one plate. I'll remove the error bars there, and you can see the individual average traces of the growth. And then we've obviously got controls, so I'll just outline what those are. So if we expand that bit, we've got two controls. We've got one where the strain we're trying to kill is just grown in 198 microliters of brain heart infusion broth. And this is the E. coli screen, so plus 2 microliters of E. coli culture. Okay, that usually grows the best. But we've also got a second control, which is 98 microliters of BHI broth, plus the filtered supernatant, because obviously when things grow, it's going to use the media. So we sort of mimic what we are adding in terms of the cell-free supernatant in that control. And then that means we're interested in anything that comes below control 2. And it also means that our interest is proportional to the difference in the controls. So we've essentially got what Top Gear used to have with the fastest time around the track. Okay? If we get a new strain which has got a lot of activity against a particular pathogen, that goes right to the top. And then we deprioritize everything else, because we want to be able to finish fast with something which is really useful. We want to find that data quickly. So the keen-eyed amongst you may have seen that flat line on the previous graph. That was quite exciting. That was from Jackie's external and very old coal scuttle, okay, which is nice. And if we have a quick look at that, what we did, this is the coal scuttle. We showed that the cell-free supernatant assays were repeatable in terms of being able to completely kill E. coli. The bioinformatics of the whole genome showed that actually what it was is a peanut bacillus polymixer. So this is the natural producer of polymyxin. The biosynthetic gene cluster for polymyxin was there at 100%. We then ran through the fractionation of it because we thought, okay, this is known, but actually it'd be quite useful to have a natural producer of polymyxin in the lab, because we use polymyxin quite a lot, it can be quite expensive. So we fractionated it. We got the fraction, which shows activity. It's a single fraction, which is quite unusual. Usually you get a bell-shaped curve as the purification sort of spans over multiple fractions. That fraction, really good, killed it. We tried it on multiple strains, some of which were colistin resistant. Those grew, so we're pretty sure it's colistin, and it's just in Swansea Centre for Mass Spec at the moment, and we should get the confirmation that we've got a natural producer of polymyxin, maybe today. No, not yet. It could be today. And that really validates the entire pipeline in that actually we don't want to, but we have replicated the discovery of a really useful last resort antibiotic. We're just about 55 years too late. But the next one, you never know. Okay? Now, you may have been thinking we've got all these bacterial isolates. What do you do with them? So we've got every single one in the microtiter plates in the freezers. So this is quite a unique microbiological resource. It's probably one of the largest environmental microbiology collections, certainly one of the most diverse due to incorporating the public into randomizing sample collection. And this will probably form the basis of a small medium enterprise company that I'm going to spin out in a year or so, offering this resource for other customers to screen for biological activity that they wish. It seems pointless it's sitting in the freezer. There's no way I could screen it for multiple careers. So it will never be screened at full utility unless other people screen it. So we just have to be able to fund that. So by rolling it out as a company, I think that is one way to do it. Now, An important thing that you may have realized I'm going on about a lot is money. For sustainability of any project, you need funding. It's just the hard truth of what we do. It costs a lot of money to pick up a pipette in a lab and then throw away a tip and get it autoclaved. So I was just going to go through the funding mechanisms that I've had. So pledges from the public. In 2020, that was about 5,000. That's probably doubled now. Pledges from grants and events that we've done, probably around about 10,000 now. So that's quite nice. It's enough to keep going. I got a prize, NIHR and UCL Research Prize at the end of 2015. That's 20,000. So that got pumped into this project as well. Kept it going. Institutional support is priceless. Okay? So really work with your various teams within the institute to prioritize the project and make it strategically important so it's pulled in at every single opportunity, not just by you who's running the project, but by your institute as well. There's a small bit of BBSRC funding where we are still actually looking at that loss of activity that I mentioned. And then we managed to get a capital grant from the NIHR, which paid for the multi-plate readers that I showed. And that was really good because that increased our capacity, sort of being able to do hundreds of growth curves overnight. That one is a charity for a new incubator. There's an educational grant there. But you can see these are all quite small fry. But just by luck, what happened in 2021 is that we also got Strength in Places funding. And this was to fund the ICON, the Infection Innovation Consortium, which LSTM now lead. And within that, we put the Swab and Send library as a kind of a progenitor for what we thought would be a bigger library, and we managed to get £3 million worth of funding for that. And one of the ways that we did that is that we valued the resource that we'd already built. So if you were to go to the NCTC and ask for a strain, that would probably be about £100. Okay? We had 20,000 strains at that point, times £100. We've got £2 million worth of leverage, so 3 million is not actually that much compared to that, which is nice. It's all words, grantmanship, but it works. Okay, so what we're going to do with that is, what we're doing now is expand the natural isolate library into a modular resource, which will be available to industry when we roll out. And we can now provide full fractionation capability. So we use the Strength in Places money to get £250,000 worth of fractionation kit. And we can now go from swab to compound ID pretty much in-house, apart from the last mass spec, within a month if we prioritize that. And that's quite a unique offering. And this also continues our work looking for anti-Candida and anti-E. coli activity until at least 2025, which is a nice position to be in. So to conclude, with your own project, clear messaging and aims and reiterate them over and over again. It's got to be a mantra. That's how people will recognize what you're doing. You need to ensure sustainability of funding and interest. Think long term. I never planned when I came up with Swab and Send to still be talking about it in 2022. It really was like, this would be quite nice for a year or so, but it's the project that can never die. I've tried to make it die early on, and people kept sending me checks and asking for swabs, which is great. Be inventive, okay? You probably can't plan everything, but seize on those opportunities and align with collaborators and other projects which are synergistic. The payoff is usually a lot better. And importantly, have fun. It can be an incredible journey. I mean, it's brought me to Northern Ireland. I think it's fantastic. You know, you've got to like what you do. And I think if you're having fun in the project, you're more likely to incorporate it in your day-to-day routine and rather than viewing it as work. It's kind of a hobby. And then my final insight is about the team, okay? Obviously, I'm not doing any of this work in the lab now. I'm just wandering around talking about it. But the people who are doing the work, Claudia, Amy, Ellie, and Annabelle, they are fantastic at this, and they really own the project. And I would advise you to really consider, in your appointments to the project, enthusiasm over skills. Skills can be learned, skills can be taught. It's that enthusiasm which is key to the success of the project. And with that, I'll be happy to take any questions. Thank you.
Well, I think you all will agree with me that that was a fantastic talk. Can we give Adam another round of applause, please? So we can ask Adam some questions now, maybe 3 questions, and Adam will be around, and after that, we'll carry on. Mm-hmm. Any questions?
Yep. Thank you.
2 sort of mini questions. What sort of metadata do you get associated with your samples that you get sent to you, and are the genes that you sequence available to
Okay, good questions. So the metadata when we send out the swabs, we send out an information sheet as well. We ask for minimal data, basically just what it is. But the metadata we get back can vary. So it could be shoe, or it could be a shoe which has been on the left foot of myself, and I'm 6'3", and I'm 10 stone, and I walked around Ben Nevis for 5 weeks, and some people really like giving you a lot of information. And we're very agnostic about it. So realistically in the lab, we don't care where it's from. We don't even care what bacterium it is. Once we know what molecule it's producing, we would then be going and talking to our synthetic chemists. Can you make this? And then the microbiology aspect is gone. We would get derivatized series of those, and we'd be testing them in the lab. So while it's fun for the citizen science project itself to have all that metadata, it's not necessary for us, so we don't demand it. People are usually quite enthusiastic. I think if people pay 30 quid for 5 swabs, they really want to tell you what they're swabbing, actually, because they're invested into it. In terms of the genomes that we're sequencing, they are coming online, freely available when we publish, just in case there's something in there that we can't reveal at the moment. And there are a few which we wouldn't reveal at the moment, because we still don't know what they're producing. Again, those samples are with Swansea.
Any other questions?
Thank you very much. Yes, you are in a way to discover new antibiotics.
Mm-hmm.
What do you think, and this is no one or new antibiotics, not the book very potent in terms of the whatever bacteria, but do you think how these will link to the resistance development? Would this be the next type of silicone clones, other drugs already developed, microorganisms already developed resistant to them? So do you think this kind of process of finding these drugs or small heating chemicals will be the same in maybe few years, and sooner or later we also go into the same pathway of antimicrobial resistance development again?
Yes.
So what are you going to do?
So we're going to find more than one. Okay.
So you're not stopping, just going to this one. Is any other way, would there be any other way you can find something with less tendons of resistant development?
Yeah. So I'll make a couple of comments on that. I think they're very valid points. So what we're looking for is novelty. Okay, and that's primarily to try and de-risk any existing resistance mechanisms which have already been selected for by our current use of antimicrobial agents. So if we find something which is producing something that we know, we deprioritize it, move on to the next one. When we find something that we consider novel, one of the first things we will do is look at resistance generation. So we've got, I've also constructed a large series of metagenomic libraries from various things where we look for resistance against novel antimicrobials in order to see if there are genes in various ecosystems which, when expressed in the target organisms of the novel antimicrobial, could confer increased tolerance or resistance. So we'd go through that process as well. Anything with a high level of resistance already present in the environment, either in this particular sort of platform of libraries or just simply by mutation, we would then be testing derivatives of that to see if we can bring that mutational frequency down, if we can make it a little bit more sort of resistant to resistance development. So those things have all been considered and put in to our workflow. In terms of things that you could do to try and minimize it, I don't think we would find anything in our assays, because that's not quite what we're looking for. But I am aware, for example, of some compounds which can modulate the mutation frequency of bacteria. And that might be good to use as an adjunct to existing antimicrobials, all novel ones that come through here, in order to prevent those from generating resistance to the antimicrobial agents that we're using. So there are strategies, but I'll just say one final thing on that. The development of resistance is an absolute inevitability. Okay? It's just we've probably got different windows of time in which to play. Yep.
So how do you comment on the past 4 decades there's no new novel class of antibiotics being developed by the pharmaceutical companies?
Okay, so there's no new classes of antibiotics which have been brought to market. There are new classes of antibiotics which are in development and have been development and then have been abandoned. And they've been abandoned for one of 2 primary reasons. One is that the companies developing them have gone out of business because there's no real funding for it. And the reason for that is that there's no profit in antibiotics. It's my view that nor should there be. There should be public goods. We should pay them centrally from government. We could talk about that later. But also, if you do finally manage to navigate all the different hurdles of bringing a new antimicrobial to market, and it's a long road, it usually costs about $1.4 billion US. Once you do that, your company is worthless because you're not going to sell enough to recoup that $1.4 million. So it basically goes bust, which is an absolute tragedy. So how that can be fixed, I think, well, there are multiple ways that can be fixed. Delinking sales from revenue. So the UK, for example, is trialling the subscription model. I don't really agree with the way they're doing that. The 2 drugs that they've chosen to invest £10 million a year in over the next 5 years, so that's quite a lot of our taxpayers' money. Those were already approved before the subscription model came out, which is great news for Pfizer and Shionogi. But we were already buying those antibiotics at a cheaper price. So, you know, where's the innovation that it's going to capitalize on? I'm not sure. But there are other things in discussion in Europe, and there's the Pasteur Act in the States, and I think we should try all of them. They should not be mutually exclusive.
Okay, one last question. I know we're running out of time. Yes?
Given that access to microbiology in those sort of settings is a huge barrier, I love your chicken gravy. Anything else? Novel, soup?
Well, I think we can all go back into our kitchen cupboards, really, and start bringing stuff into the lab. But it seems like an underutilised resource. I have to say as well, that chicken gravy was out of date, which is why I looked at it. I thought that's quite far out of date. What could go off in it? And nothing could. But, absolutely. Yeah, I think it's something that could be utilised a lot more. It saves a lot of money.
Thank you very much again, Adam.
Okay.
So for the interest of time, I am going to leave the next item to the last. So I'll go forward and then I'll come backwards, okay? So that next item is a kind of representation of all of the people who have signed up for the AMR Hub. So far, the hub launched about a month ago, roughly, and we have almost 70 members signed up to it. So if you haven't signed up, please do sign up to it, because you'll be hearing a lot more about all of this kind of stuff, and hopefully we will come together and make something great out of this. As we've heard, interdisciplinary work is paramount. Also, I just wanted to use this opportunity to thank the Microbiology Society, the School of Biological Sciences, and the Institute for Global Food Security for supporting this event, and the AMR and One Health Lab here as well. So that's why you can see some posters. They've all been really instrumental for this. So I'm just going to skip that there, and I'm going to go straight on to the next slide, if it lets me. It may not let me. Oh, there we go. The Great Cat Project. So I came up with this in the middle of the night when I had COVID. You know, like when you can't sleep because you have COVID and you have temperature. Sometimes they say when you have a temperature, you have hallucinations. So it may have been an hallucination. I don't know. Because I've always admired and been inspired by the Swab and Send, and I've always wanted to do something to do with citizen science. I'm very involved with outreach here in Northern Ireland, and I go to schools. Last year, we visited 40 schools here and reached almost 2,000 children with hands-on microbiology work. And I've always thought, how can we make this more sustainable and incorporate our research into it? So as part of the AMR Hub, please, this is a job for all of us, the AMR Hub, because as you've heard today from Adam, I can't do it by myself, okay? So the Great Card Project essentially means the Great Citizens Antimicrobial Resistance Together project. Now, that's your job to help me find a snappy mantra, like Adam have said, if that doesn't make any sense to us. Because I thought CART means that it can carry people, it can carry a lot of people, and I would try and find a better logo for that, obviously. That's just me playing around with Adobe. And so what's the goal of the project? We want to raise awareness about AMR because we need continually raise awareness, but we also want citizens to be able to contribute to the research and feel like they're part of solving the problem through sending us samples and being involved. And why? Because AMR is not just a scientist's problem, it's everyone's problem. It's a threat to all of us, like we've heard today. I'm not going to go into that. So it's a truly global endeavor, and it's a fight for everyone, that we must all join forces. So we have to stop playing catch-up with AMR. That's one of the goals for the Great Cat Project. What we tend to do is wait for a microbe to become resistant, or we isolate something from a patient or from an animal or from the environment that is resistant and has caused an infection, and then we try and play catch-up. Oh, what's that microbe? What's that pathogen? What's the resistance? And then we try and find what can cure it. I think it's high time we stop playing catch-up, and that's where the Great Cat Project comes in. So why not find out as much as we can all the resistances and the phenotypes? Look at all these methods that people are using today, as we've heard. Why not try and characterize as much as possible all of the resistances in all of the available microbes that we can grow, whether from the environment and otherwise, and then hopefully that would help inform quicker diagnosis later on, and we wouldn't have to be playing catch-up if somebody accidentally picks up an opportunistic pathogen, for instance, from the environment, etc. So that's what the project is about. We will officially launch this project during the NI Science Festival, so there's a program already for that. So please join Twitter, and we'll create Twitter accounts as well, and please just help us get this out. We want people to sign up and contribute to the project, so we will make kits. I'm going to be talking a lot with Adam because, he's the guru of this. He's a veteran. He knows what he's doing. And people will send us swabs, or we will have collection points, especially on the day of Science Festival. We'll have the collection point here. And then we will try and grow these isolates, just like Adam has been doing, or use genome technologies to try and characterize the resistances. So we're hoping that this will also bring some funding and try and find funding modalities around it, as well as studentships and so on. So that's what the project is about. So volunteers, everybody who's going to join me, please, everyone, and we can do this together. So thank you very much. It is now time for you to network some more and have some posters. I just want to take the opportunity to thank all the speakers who've made out time, all of you who've come today. I hope you've enjoyed it. And I'm going to take the slides back to the beginning so you can sit down, relax, and look at all the different things that people are doing. It's like a little video scroll. All the things that people are doing around AMR, not just within Queens, but in Northern Ireland as a whole. So lots of people have signed up for this. So thank you so much.